Hello, my name is Federico Baruffi and I'm going to present the contents of my PhD project, Integrated Microproduct Process Quality Assurance in Microinjection Molding Production. This project was carried out under the umbrella of the European MICROMAN project, and it was carried out also at the Technical University of Denmark, DTU. The main focus of the project was on the manufacturing process called microinjection molding, which is the micro-scaled counterpart of conventional. The two processes, even though they originated from the same principles, are different. So a special machinery is needed to carry out the micro process, where a plasticization screw and an injection piston have to carry out the process. Micro-tooling processes have to be used to manufacture the micro-scale cavity, processes such as micro-EDM and micro-milling, and finally, peculiar physical phenomena happening only in the micro-scale are appearing in micromolding, such as wall slip, for example. So there are differences between the microscale and the conventional size process. Quality assurance is one of the main drawback of microinjection molding, one of the biggest difficulties, because if we follow the conventional process chain, we have the throughput of the process is very high as a fast verification process. We have typically more than 50 parts per hour, but the offline methodology, which is applied to measure the parts, has a very low throughput of typically lower than 10 parts per hour, because we need to employ a very precise 3D instrument to measure a very small part. So there is a mismatch, and typically only few parts are selected randomly. On the other hand, we have the fingerprint scenario, which is the fingerprint that was applied in the PhD project. And we start from the same throughput, so the process itself is the same, but we apply process monitoring to carry out quality assurance, and monitoring is carried out at the same speed as the process. So we can have actually information on all the parts that we produce, but we need to study the correlation between the monitored curves and the quality of the parts. This was done based on the product process fingerprint approach, which relies on two definitions: the product fingerprint, a dimensional outcome that is sensitive to process variation and correlated to overall quality of the micropart. This is a product characteristic that, by measuring it, can give an idea on the overall quality, just by measuring a single characteristic. Then we have the process fingerprint, a process variable strongly correlated to the quality of the produce component and that can be controlled at each cycle by a monitoring technique. So by merging product and process fingerprint, we can reach the total quality assurance that would increase the quality of the process in a very sensitive and consistent way. The case study that I'm going to present, on in which we apply the fingerprint approach, is based on antibacterial application based on a nanostructure surface, which had produced by a picocycle laser and having ripples of 50 nanometers in height and 100 nanometers in pitch. And this is the SEM picture showing that also the surface as a single layer. The monitoring is very important, as I said, to achieve a total quality assurance. And the setup was a single cavity, two plate mold, having the monitoring as an infrared camera, high speed camera providing information on temperature through infrared and also on the shape of the flow itself. This was carried out using a 45-degree tilted mirror in the mode that allowed the visualization from outside. Experimental design was based on the micropower 15 machine, so a injection molding machine dedicated to the micro scale, and carrying out a 2 to the power of 3 full factorial plane in order to analyze the effects of three parameters: holding pressure, injection speed, and mold temperature on the replication with two materials, POM and PC, so polyoxymethylene, a saline crystalline material, and PC, an amorphous material. So by comparing the data on the two materials, also an idea on how the replication works with different molecular structure could be achieved.